Today, let's try to understand Penicillin G versus not versus, just Penicillin V, right? These are the first beta-lactam antibodies, and the mechanism and function of these penicillin are is just they have something called as beta-lactam ring. Now, this is the core structure of the penicillin, and what happened here is this ring, right? This resembles something of DLA, right? And now this structure called DLA, this is the normal substrate that bacteria uses for the formation of the cell wall, right? So basically, what penicillin is doing is it's just doing this mimicry, right? Now, because of this mimicry, the penicillin is tricking the bacteria, right? And because of this mimicry, tricks the bacteria and enzymes, right? This is how the whole penicillin works, right? Because it has something called as beta-lactam ring, whatever you want to call it, and it's a DLA ring. The PBs, right? What are PBs? PBs is penicillin binding protein. Now these PBs, they link means they link like that. They link something called as peptidoglycan chain. Now these peptido means protein, glycan means sugar. So sugar protein chains together. They link these chain together, and they do this by cross-linking, right? And this cross-linking, especially this cross-linking, is the strength of the bacteria. Now what happened is here is this penicillin binds to this peptido binding protein, right, irreversibly, and it prevents the linking. So for example, this is a wall, right? And I'm gonna make a wall right here, and this is the wall. What happened here is this penicillin actually puts a brick right here in the middle of the wall, so that the wall cannot be made successfully. Once you put this brick inside in an odd position or in a incorrect position, you won't be able to make this wall. So bacteria cannot bind strongly, cannot build a very strong wall. So that's the idea. After this, what happens that if you have bad walls, you will have something called osmotic lysis, of course. Now, I think we really should really try to understand why osmotic lysis is really happening. What normally happens in bacterial cell wall synthesis is that bacterial cell wall is made up of these peptidoglycan, right? And this peptidoglycan is a glycan chain, right? It's cross-linked by small peptide bridges, right? And the enzyme that makes these peptide cross-linkings, it's called penicillin binding protein, right? This one, PBBs, right? And it links these chains, right? So what happens is this penicillin actually, it mimics this, this this peptidin binding protein. It mimics this DLA end of the normal peptide substrate, right? And what happened after this, that PBB, right? It tries to react with penicillin, and it tries to tries hard to form a covalent and a very irreversible bond with it, right? So that makes this penicillin binding, that makes this PBB, right? This PBP really bad. Like, it cannot cross-link now. This PBB is inactive now. Now the result of this is that they have bad cell wall, they have defective cell wall. Now bacteria, they try to keep it, they try to make it again and again, but their cell wall is really weak and fragile. After, what happens is that the cell wall what happens is that the existing peptidoglycan still is there, right? And the cell tries to grow and it tries to divide, so it must insert new cell material, right? Or it can remodel the old wall, right? Now during this remodeling, or during this growing stage, the bacterial has something called as autolysins. Now these autolysins are these bacterial self enzymes that cut the old wall and become active, right? Now normally autolysin activity is cell wall synthesis happen, and the cell wall synthesis rate and the autolysin activity is balanced usually. But now the cell wall synthesis is messed up, and autolysins activity is normal. So autolysins is just working, and autolysins keep cutting, but there is no new cell wall. So what happens is that this autolysin is the reason for this bactericidal, right? The bacterial cytoplasm has really high osmotic pressure than the surrounding medium, right? And what happens is this, what's happened here is what happened here is that these peptidoglycan layers, right, these layers, the plasma membrane, it bulges out, right? Right now, when the bulging is happening, this is irreversible. This is reversible damage, right? But when the cell burst, right, this is irreversible, because now the content of the inside of bacteria is outside, right? Now it cannot, now it cannot reversibly get, normal. So the osmotic lysis happen, and this is exactly what osmotic lysis is, because the pressure inside the cell, right, inside the cell of the bacteria, it's so much that the bacteria, it bursts. So this is what exactly osmotic lysis is. Okay, so penicillin is a bactericidal because it stops the cross-linking. This is stopped. It and the auto license that bacteria always that bacteria ooze have, that bacteria already have. They are normal, right? They don't have any down regulation of the auto license, so the cell ruptures, right? So this is what osmotic lysis really means. The spectrum is mostly gram-positive bacteria because they have good cell wall. They have good cell wall that is made up of peptidoglycan, and the medicine can mimic DLA substrate. Types is, we have one type that is V and another type that is G. So I usually remember V as vinegar, which is acid stable. It means you can give it orally, right? Our stomach has acid, so it's poor bioavailability, but it's great for oral. It's great for oral, and it's great for minor infections. You can give it IV, and it's more powerful, of course, because it's IV. And this type is called G, and we can treat streptococcal pharyngitis. That's the first line for streptococcal pharyngitis. We should give penicillin. RH fever, rheumatoid fever, strep viridans, endocarditis, GBS, group B, streptococcus prophylaxis in pregnancy, because these are all peptidoglycan cell wall bacteria, right? Clostridium perfringens, it can give you gas gangrene. Other thing, Postrella, that is due to dog bites, syphilis, and Neisseria meningitis. So these are these things. And actinomycin, what is this? So right now I'm reading about this actinomycosis. It is caused by something called as Actinomyces israeli. It's a cool name. And what happened here is it's also a gram-positive. It's an anaerobe. It doesn't need any sort of oxygen. It's a non-acid fast bacteria. It's not a fungus, right? And it just lives normally in your buccal cavity, in your oral flora. So what happened here is that they cannot penetrate right inside the mucosa. That's why they can usually live in your buccal cavity normally. What they usually do is that they kind of they do some sort of they can get inside your body by, if you have some sort of gum problem, if you have some sort of tooth infection, if you already have some abscess, or maybe some sort of poor oral hygiene, right? So once your mucosa normally breaks because of your bad habits, right, poor oral hygiene, the actinomycins can enter inside your submucosal tissues, and they don't need any oxygen, right? So they thrive, and so they can affect. So this is how it works. And what about this jaw abscess and sulfur granules? Well, inside the tissue, the bacteria grows really slowly, and they can trigger inflammation, and then the pus happens, right? And then what is pus? Pus is just fluid with dead neutrophils, and then a lot of pus is happening. So this gives jaw abscess, right? And what about sulfur granules? Well, what are sulfur granules? First of all, they are just pus from the sinuses, right? And they contain these yellow granules, right? They are called sulfur granules, and they contain actually no sulfur. The name is just sulfur. So they are just filaments, and they are yellow, and they are like sand grains, and you can see them under a microscope, right? And that's how they look like. That's it. So they are just filaments of these bacteria. So that is sulfur granules. And so we can treat these conditions. Side effect is type 1 IgE allergy, right? Rash. So anaphylaxis, of course. Hemolytic anemia, that is positive Coombs test. So positive Coombs test is like detection of antibodies that attach and destroy RBC. Coombs test is like if RBCs are getting agglutinated or not, right? That's the concept. So when I'm trying to say agglutination, right? What is agglutination? Agglutination is just clumping of RBCs, right? Due to antibody binding to their surface antigen, right? So antibodies, maybe IgM, IgG, maybe, I don't know, anyone, I don't know. It's an or IgM or IgG. It binds to the antigen on the surface of RBC, and these antibodies, they act as a bridge. So that's how this glutination happens. So that's the concept. Interstitial nephritis. Interstitial nephritis means it's an IgE mediated. It comes under the type 1, and it's just kidney inflammation. That's it. Resistance of penicillin G and penicillin V can be if any bacteria have something called beta lactamase, and it's also called penicillase. What happens is that it can break down, or it can this cut lysis of beta lactam ring that penicillin has, and this usually beta lactamase come from something called as plasmid gene. This carries this resistance in themselves, right? Now this plasmid gene is a small circular double-stranded DNA molecule found in bacteria, separate from their own chromosome DNA. So it means it is something which they have learned. So this is plasmid gene. So any bacteria that has this plasmid gene can actually can be resistant to penicillin. So that was penicillin G and penicillin V. I hope you have a nice day. Bye bye. Take care.